Hello, I'm Giacomo Sferruzza from San Raffaele Scientific Institute, and today I'm going to talk to you about transcriptional changes induced by fingolimod on T and B lymphocytes of relapsing remitting multiple sclerosis patients. We selected a cohort of 24 patients treated with fingolimod in our center, and we didn't include patients treated with immunomodulant or immunosuppressant in the 3 months before or natalizumab in the 9 months before. We collected blood samples at baseline and after 6 months of therapy, and we obtained B lymphocytes and T lymphocytes from them and performed a transcriptional analysis on them. We performed a statistical analysis in 3 different steps with 3 different aims. In the first step, we basically look at the differential expression for each gene. In the second step, we performed a pathway analysis looking at the pathway that were enriched by differential expression genes, and we used an innovative tool that is called MainPath that basically put together genes using Boolean operators that was performed take into account the functional relationship between genes, creating these strings that are called subpath. And we basically study the activation, the coherent activation state of these subpaths at baseline and after 6 months. In the last part of the study, we performed the network analysis, putting together the data about differential expression and the centrality of each gene. Our study was not performed at the single cell level, but it was a transcriptional study of an entire population of B cells and T cells. And as we know, fingolimod can induce a strong change in subpopulation amounts of these 2 big populations of T cell and B cells. So exactly as we expected, we observed an overrepresentation of genes specific for cells that were already in the blood after fingolimod treatment, and a down-regulation of genes specific for cells that were trapped in the lymph node by fingolimod, like T central memories, for example. Most of these genes were related with the cell migration, obviously. The results that we didn't expect regard the effect of fingolimod on immune function genes, because what we observed was an overrepresentation, an upregulation of several genes related with the pro-inflammatory profile of the immune system. And it was the same results that another study performed on an independent cohort in Germany observed last year. What we performed more than this study was a network analysis in which basically we observed a downregulation of several central genes that suggest a lower activation state of these lymphocytes. So, in conclusion, what we observed was a strong modification of genes related with cell migration that can due probably to the change in subpopulation subset of T lymphocytes and B lymphocytes, and the cell that was spared and that were already in the blood after fingolimod treatment seems to have a more pro-inflammatory profile, but associated with a lower activation state. Well, obviously what we have to do, the next step, will be to have a functional confirmation of our results, and the final aim of this study, that will be the second step, will be to look if in this transcriptional modulation there is something that could predict the long-term response to fingolimod treatment. I'm Baveno this year because I'm really interested in the topic of clinical study with a long-term observation, because in my future I'd like to study the personalized therapy for MS patients.